And we're now welcoming our last speaker before break, who is Nosarieme Abey, who will present a talk entitled Dietary Consumption of Cannabis. Sativa alters cognition, neurochemical balance, and cellular organization in Sprague Dawley rats. So, we can see you now. Can you start sharing your presentation, please? Yeah, perfect. And I let you talk now.
Thank you very much, Valentin. Greetings. Hello, everyone, and welcome to my session of presentation. It's a great pleasure to have this opportunity to present in this virtual conference, and I want to say a very big thank you to the organizers of this conference for giving me the opportunity to present my work. I will be speaking on dietary consumption of cannabis sativa and its alteration on cognition, neurochemical balance, and cellular organization, and Sprague Dawley rats as a model for human. Cannabis is a plant, and its major occurrence is a sativa or indica, and this could be in the wet form, the fresh form, which is green, and or dried form, which is usually brown in nature, and it depends on the period of presentation or where it is being represented. It goes by different names like weed, herbs, pot, and the likes. And all forms of cannabis is capable of altering the way brain functions. And this is because it is highly neuroactive. Scientists have been able to identify a number of hundreds of chemicals as a component of cannabis sativa, but the most psychoactive component is the tetrahydrocannabinol. 2.5% of the world makes use of cannabis, and this use could be medical or recreational. Of major concern is the recreational use, which in most cases is unintentional, and it can lead to excessive use of the drug because it is not controlled. And sometimes extracts from this plant are made into different products that are consumed without necessarily having it as drinks or biscuits, and the dosage of use is not known. So when it is consumed orally, which is a point of focus for this research, it takes longer time in the body because it's able to penetrate to different parts of the body, unlike when it is smoked. And how long it stays in the body is a factor of the tetrahydrocannabinol, which is the active ingredient in this plant. And the function also of the fats deposit in the body of the animal, and the size of the animal, the frequency of consumption, and the amount of the plant that is being utilized per time. There is an established endogenous cannabinoid system that modulates the neuronal signaling in all animals, and these are called the endogenous cannabinoids, and of major identity is anandamide. So what tetrahydrocannabinol, which is an active ingredient in the in cannabis sativa, does is to mimic this natural neurotransmitter, directly or indirectly flooding the circuit with dopamine, and so activating the reward system. And this is what gives the eye feel. This is what makes the animal to feel high when they take the plant. Because it is a structural analog of the endogenous cannabinoid, has almost the same structure, is able to compete with it and bind with its receptor, which is Cannabinoid One Receptor, available in different parts of the brain, causing the downstream endogenous neuronal signaling, which include neuroprotection, neuroplasticity, and the likes. But when tetrahydrocannabinol binds with the CB1 receptor, it disrupts this normative endogenous neuronal signaling and causes its own modulated downstream effect. Cannabinoid 1 receptor is a major receptor that tetrahydrocannabinol targets in the brain, and it alters the function of different parts of the brain where this receptor is found. One is movement, which is the basal ganglia, coordination at the cerebellum, memory at the hippocampus, The nucleus accumbens at the reward system, and judgment at the parietal cortical region of the brain. And I'll focus on this work in a bit to understand the neuronal neurological effect of consuming cannabis in the diet. We identify some key neurotransmitters which are very key to learning and memory, and we checked for the assayed the level and also the overall radar status of the brain. And this we did by getting the standard rat chow, which is required for the maintenance and growth of rats. And then we included some ration of cannabis sativa. The high dose group received 10% cannabis, the low dose group received 5% cannabis, while the reference group, which served as the control, received 0% cannabis in their feed. And this was given to them all through the period of the research. They were subjected to cognitive function tests, which involved the use of shuttle box. This box is a training box that involves a form of passive avoidance behavior involving negative reinforcement. And in this case, negative reinforcement is a food shock. So there is a conditional stimuli, which is a light bulb on the light compartment, and a wired network on the dark compartment, serving as the negative reinforcement or the unconditional stimuli that is expected that the rat learn to associate the relationship between the unconditional stimuli and the conditional stimuli, as well as bringing up a particular link between the unconditional stimuli to stimulate a conditional response, which is the cognitive response or cognitive latency that we will record. So we also did some biochemical indices measuring the overall radar status of the brain, and then quantification of the level of serotonin and acetylcholine, which are key to establishing long-term potentiation required for synaptic plasticity, which is implicated in learning and memory and retention of acquired memory. So histological representation of some of the organs that are targeted by this active ingredient of cannabis was also carried out, and the blood chemistry representing the overall status of the health of the animal was done. From our findings, at the beginning of the research, we discovered that the low dose group and the reference group were almost the same in their activity, in their physical activity, but as time of exposure progresses, the low dose group were hyperactive. They had heightened alertness and they were very aggressive, and the high dose group had a calmness and a slowed spontaneous movement, which has been likened to a catalepsy. And this shows that there is a defect already in the motor control unit. The weight of the high dose group reduced. The rats had experienced a reduction in their weight as against what we have in the reference group, which increased in weight. And this shows that there is an increase in metabolic turnover, which further emphasized by the soft defecation and the number of frequency of defecation that was observed. The cognitive response that was obtained from the cognitive challenge to which the rat was exposed to was very high. The time for the rat to execute the function and to transcend the information, process it to comprehension, and execute the function was high. This shows that there's an overall deficit in the cognition of the rat as a result of the exposure to the plant or the dietary consumption of the plant. Neural circuits that produces or makes use of acetylcholine have been implicated in cognition, and they are very key to cognition. And so there was a drop in the acetylcholine level as well as the activity of the metabolizing enzyme, which is acetylcholinesterase. This shows that the use of the cannabis sativa is able to modulate some of the normal influences of acetylcholinesterase on the normal physiology of hippocampal and cortical dependent memory association. And because it is very key to cognition, the establishment of long-term potentiation, which is required for synaptic plasticity, is disengaged, because there is need for a strong firing, a strong communication between the neurons for long-term potentiation to be established. And so this also applies to serotonin, which is capable of altering part of the brain network through different parts of the brain, the hippocampus, the amygdala, and some other parts through modification of synaptic plasticity. And this it does by moderating cross-modal plasticity, a procedure that is essential for cortical restructuring and powering synaptic plasticity through the adhesion complex that is responsible for the formation and function of neuronal circuits. The drop in this level of key neurotransmitter is a very fundamental input that we have to take note of, because it will underlie so many other pathophysiological brain disorders which could creep in as a result of the use of the dietary consumption of cannabis sativa in the rats as a model in this case, in this research. So the overall radial status of the rat was overwhelmed, although it has to be noted that at the beginning of this research, the low dose group even had a better antioxidant capacity compared to the reference group. This shows that the medical use of cannabis sativa could actually be established if it is controlled and under a dosage that is of unique control. And that is what medical use is all about. But in the high dose group, it was highly overwhelming, and this shows that the condition of the brain is already stressed. Liver, which is the major organ of target when it is consumed in diet, because of the first pass effect, was also disrupted. The cytoarchitecture is disrupted because some of the biomarkers that shows that there is a leach from the organ were very high in the blood, and this also altered the overall blood chemistry of the rat. The histopathological examination of the brain, the testes, the liver, and kidney further emphasized the alteration in the cytoarchitecture as against the control, especially in the high dose group. The low dose group never had any alteration in them, but in the high dose group, there was alteration in their cytoarchitecture. And for example, the testes, there was vascular congestion, and this supports the physical activity that was observed in the case of those animals in their arousal to opposite sex. So there is a part of when cannabis is consumed by rats, the neuronal behavior is altered and the cognitive function was also altered. And this was underlined, though dependent on dosage and the length of administration. And this is underlined by reduced antioxidant capacity of the brain, causing a stressed condition in the brain, and also an altered level of serotonin and acetylcholine in the transmission of neuronal plasticity that is very key to long-term potentiation required for establishing a synaptic plasticity in learning and memory. And so some of the communication between neuronal components are weakened, and so there is no firing of signals, and so there is the ability to retain or recall what has been learned is very weakened. The weight losing effect is already established, and this has been linked to the components tetrahydrocannabinol that increases the metabolic turnover in rats. And so we conclude that having rats, which has been established to have almost the same drug-seeking effect as human, consume cannabis can lead to a dose and time-dependent cognitive function deficit with reference to altered functional level of neurotransmitter system and radar status. And these are serving as the underlying neurochemical mechanism for this effect to take place. So this contributes to a body of knowledge on the mechanism underlying the cognitive deficit that cannabis sativa consumption in animal model, specifically rats in this case now, embarked on to be able to affect this neurological changes. So thank you, and please stay safe.
Well, thank you a lot for a great talk. While the chat prepare his question, I will let Alexis start the first one.
Yes, thank you. We could not really establish that, but we use that sound as an unconditional stimuli that will trigger that effect. And so I think, and from what the research shows, it shows that their hearing ability was already altered because the air is linked to different parts of the brain. And so because it's all, as well for hearing and balancing, and so because there's an alteration in some parts of the brain, it could be possible that this has also altered some of the hearing signaling processes as well.
I have a first question in the chat for you. So Agvashiki Obungadu is asking, is it possible to control the use of cannabis by people based on your low dosage finding?
And that is why, like I mentioned, the recreational use is of great concern because it is not controlled. It is only when it is used medically by a controlled setup that you are sure of the dosage that you are being administered to per time. So controlling the amount people will use might not be sacrosanct, if I can put it that way. You can't be too sure because this same substance is also addictive. It has a way of wanting you to get more. And so when you get exposed to it in the first instance, maybe for a medical purpose, and it is not under a control, you want to have more and you want to have more, and then you get to abuse it. And then it gives its destructive and downstream effect on you.
Thanks. The second question from Estela Braga Nepomuceno. Great talk. I would like to know, what was the age of the animals when you start the diet?
They were around, because we focused on the adolescent group, which that was from 6 weeks to 8 weeks. The average of 6 to 8 weeks was the age of the animals when they started.
Thanks. Another question from Sama Abuzid. Usually, abusers tend to misuse several types of psychoactive drugs simultaneously. Do you think that this realistic condition may affect your results?
Sorry, can I get the question again, please?
Yes, sure. Usually, abusers tend to misuse several types of psychoactive drugs simultaneously. Do you think that this realistic condition may affect your results?
Okay, our focus is on cannabis, sativa. There are occasions when it comes to the human model, there are occasions when when you start a neuroactive drug, you tend to move to others because there is a quest for more. And because the reward system is already activated, there is need to sustain that heightened alertness and heightened feel of highness. So but for this research, our focus is on cannabis sativa, and that is what we're looking at, the different dosage and the time of exposure. Other drugs coming in can also further cause this downstream effect, but our focus on this research is on cannabis consumption and its neuroactive effects.
Thanks. I think we have time for maybe one last question from Sebastian Minoli. Cannabis decreases pain perception and can increase hunger. Should one expect for this reason a decrease in aversive learning and an increase in appetitive learning?
Okay. If I got the last one, appetitive learning, right?
Yes.
Okay. So the appetite is increased, and this is because there is an increase in the metabolic turnover as a result of the tetrahydrocannabinol, which is a very prominent component of the plant as well. The increase in the turnover is what triggers the quest for more food, because the ratio of the conversion ratio between the food consumed and to body utilizing it's very low, and so there is need for more because you tend to defecate more and have more of very soft defecation. But the aversive learning is a different thing entirely, because this is based on the brain hippocampal sustenance on learning. But the other being hyperphagic and all the rest is another center of the brain that coordinates that. So its effect on different parts of the brain is based on different components of cannabis exerting its effect on by binding to different receptors in those different parts of the brain. So we might not be able to give an association directly to the aversive learning leading to the appetitive learning, but it exerts both conditions.
Well, thanks a lot. I see that there are more questions, but I think we'll let them to be asked on the Discord server if you have some time to look at them there. Thanks again for your very nice talk and questions. And we are now turning to the first break of the session. So you may now leave the meeting. Thanks again.
Thank you.